Hi everyone, it's my pleasure to be here today to join virtually for the AKC 2021. My topic for today will be on polyphenolic acid hydrocolloid as an implant to address peritoneal carcinomatosis, which is a form of peritoneal cancer that is pretty common in this part of the world, in the Asian region. So I am going to give brief introductions of what are polyphenols. So for those who are really in the organic chemistry, this is something that is not very strange to you, that in our natural environment there are whole lots of organic compounds that is easily available, and these polyphenols are characterized by multiples of phenol units joined together, forming a very huge molecule. Typically, these can be found as antioxidants found in fruits and vegetables, as well as in a lot of food products such as your chocolate, your red wine, grape, tea leaves, and even in animal tissues such as muscle. There has been a lot of research done in this area, and they have been in vitro and in vivo studies that have been reported to show a whole lot of therapeutic properties of polyphenols. They have been reported to be anti-cancer, anti-inflammatory, antimicrobial, for example, which is really important in this COVID-19 era. But with the therapeutic properties that are so attractive, there are a number of drawbacks that these polyphenols are suffering from, which is the instability when they are exposed to light, to heat, for example. So a lot of work in this area is looking at how to process these bioactive ingredients in the form of stable polymers or biopolymers that will have a better stability in the natural environment, so as to achieve is, but yet to achieve is highly anticipated therapeutic. And that's going to be the essence of this talk today, where I'll be talking on the work that I've done in processing these bioactive ingredients into the form of a biopolymer gels as well as a hydrocolloid. And I'll be including in this talk some of the preliminary studies that are done on preclinical animal studies to show the potential applications of these fantastic materials for biomedical application. But before I go into that, I just want to quickly touch on a few areas which is really of my interest: tissue sealants as smart wound dressing, and finally the anti-cancer hydrocolloid as an implant. So this is a very brief intro of how we can use polyphenol biopolymers as a tissue sealant, which is getting very much attention nowadays, especially from big companies like J&J and also other big companies like Olympus. So they are really looking at this huge area of sectors. So how the mechanism works is really to trigger the cross-linking between protein and amine group that are found on the collagens and tissues, which has whole lots of collagens. So the idea here is to trigger the cross-linking between the OH groups of the phenolic acid compound with the amine groups on the collagens, or with the oxygens, double bonded oxygens from the collagens. So if this can be achieved, essentially what we are seeing is spontaneous cross-linking of the tissues, right? So this could be very attractive, because if that is something that can be achieved without sutures, that would have huge applications in the area of surgery. Okay, the next applications of this polyphenol biopolymers will be in the area, in the clinical domain of wound dressing. Conventional wound dressings, we all know that after you stick onto the skin for a few days and after your skin heals, after your wounds are gone, when you peel off your blasters, or so-called your wound dressings, very often due to the long hours of adhesions to the skin surface, very often you get complications such as blisters. Sometimes you might even get your skin tear for those elderly with very, very thin skin. We call it thin stripping as well. So here we are talking about microstructures that could be performed or could be created on the biopolymers. And with the addition of agents, of course, these agents simply just minerals to trigger the expansions and contractions of the biopolymers in order to trigger the gripping and releasing on the skin surface, right? So with this, we'll be able to repeatedly use even the wound dressing without causing so much complications such as blister or skin tear. So that's a very interesting area that is really of huge interest for many people, right? The third topic of biomedical applications, I'll be focusing on the work that I've done on the hydrocolloid. We have used quite a number of different ingredients. We have CMCs, we have PEG, citric acid as cross-linker, and dextrin, which is essentially carbohydrates or sugar. So the presence of the hydroxyl as well as the oxygen groups in all these materials as used here, many of these, they are hydrophilic groups. So therefore, with the carboxylic groups of the CMC and the citric acid, we'll be able to induce an electrostatic kind of attractions and bonding, and therefore cause external stimulus responsive kind of swelling as well. So this is what we want to achieve as a hydrocolloid. Hydrocolloid meaning that able to very much function as a hydrogel, but without the presence of water, it will be almost like a solid film. But in the presence of water or any liquids, it will be able to absorb the liquid and transform from a solid film into a hydrogel. So that is what we meant it as hydrocolloid. So the way that we make the actual products is to use something that is semi-automated machines, as you can see here. So we're using a film coater where we input hydrogel mixture into the feeder, and the rest of it is simply just pressing the button, and the machine will be able to adjust the thickness that we have preset to give a very uniform and pretty big A4 size film. So being a material, we want to know chemical properties as well as biochemical properties such as your inflammatory properties as well. So we did quite a bit of testing. We have the tensile tester here to measure the tensile strength. We measure the anti-inflammatory properties by using UV spectroscopy. We measure the swelling test as well in physiological solutions and contact angle measurements. All these are used as parameters and fit into the optimized final target technical specifications that we want to achieve in order for the film to function at its best condition as hydrocolloid in the bodies of the animals that we are going to perform as part of the preclinical study. So this is the final product after the few runs of optimization. The size here you can see is pretty big. It's 11.5 by 7.5 cm, pretty much similar to the size of the current commercial products that you can get in the market. So next few slides, preliminary results that we have achieved using this optimized film to look at the anti-cancer properties of this hydrocolloid. So what we did here is we choose CD26 colon cancer cells, which is really common cancer lines and also one of the diseases that's really common in this part of the world. So we choose CD26 and we inoculated the cell in the mouse on day 0. So we purposely tagged fluorescence to the CD26. So under the imaging, you can see that the bluish emissions would have indicated a huge presence, high intensity that's coming out from CD26 cancer cells. And on day 4, we form a surgery to open up the abdominal cavity of the mouse to implant the hydrocolloid within the peritoneal cavities, and we suture up the wounds, and we continue to observe the mouse by taking good care of the mouse under controlled environment while we monitor the fluorescence emission. So that's on day 4. So here you can see 2 groups of mouse. We have the control group and the experimental group with the implant of the hydrocolloid. If you were to compare day 7 on the left to the day 7 on the right, not much difference except we see a slightly higher fluorescence emission on the control group. But what is more important here is to compare the results at day 33. If we look at day 33 on the control group mouse and day 33 in the experimental group, you can see that the amount of fluorescence that we are seeing here is so much less in the experimental group as compared to the control group. In fact, from day 7 all the way continuously up day 33, you can see that the intensity of the fluorescence increases until day 33 when we have no choice but to all the mouse because of the spreading the metastasis of the cancer cells. Surprisingly, you can see that the mouse on the right-hand side, which has the implants in the peritoneal cavities, except the middle one, which is an exception, the other 2 seems to survive very well. In fact, on day 33, we can see a very huge drop, in fact, very minor fluorescence emission that's coming from the cancer cells, which totally disappeared as we go beyond 33 and when we move on to day 52 and day 70, when we don't see any fluorescence emissions at all, which is a very encouraging result, indicative that the colon cancer cells have been eradicated at least beyond day 52. So we just want to confirm that is really the case by looking at the ex vivo imaging of the organs. So after we euthanicize the mouse from the experimental groups, we take out the organs and we check the organs under the fluorescence imaging, and indeed we don't see any fluorescence at all, which is confirmative that all the cancer cells have been gone. So with this, I want to thank you, the organizing committee, for inviting me to this talk, and going forward we hope to be engaged within this very interesting community, and maybe we'll see each other again next year. Thank you for your listening and attention. Thank you. Bye.